Punnett square is what they use for. This is like a no-brainer. Okay, right? It's probability. So it's really to show you the possible offsprings from a cross. The top. What? We'll just break away from this for a second. In a Punnett square, we'll just. Okay. So these are the possible offspring in here. Actually, F1 offspring, right? So what are these things out here? The parents are up here. So then, what are these things? Come on, you guys know this. The gametes. So these are the gametes, or sex cells, where the segregation, so big T, little t, so the segregation separates those gametes into two separate cells. If they're eggs, and you could say it's big T, big T, little t, and then these would be sperm. We don't show them like this because it gets kind of ridiculous to draw those. But, so those are the potential gametes for each parent. Okay? That's what those are. And then these gametes, what are the possible combinations? This sperm goes to that egg, this sperm that egg, this sperm that egg, this sperm that egg. That's what the Punnett square illustrates. Okay? All right. If one of the parents is homozygous dominant and the other is homozygous recessive for a trait, so homozygous dominant, homozygous recessive, what will the offspring look like, and what will its genotype be? Let's see, who am I got? Let's go over here. Josh. What? The question is, what are they going to look like and what's its genotype be? So the trait, it'll be Austin Help.
It'll be the dominant trait.
That may have been what you're saying. Okay, so they'll all be the dominant trait, and what will their genotype be?
It'll be heterozygous.
Okay, heterozygous or hybrid, very good. Okay, that's basic stuff. You should be able to, by now, do that. Okay? All right, parents heterozygous for trait. And again, this is just seeing if you remember your vocabulary. What's the chance the parent giving a dominant allele to the child? Should be really great. No, but you can take it if you want. It looks like Grace wants to get off the hook. But
It's hard to do it. It's hard to think about it.
No, this is pretty straightforward. So if a parent's heterozygous, what's the chance of that parent? So big R, little r, what's the chance of her giving the dominant trait to her child? 50%. It's flipping a coin. Okay? This is the probability thing we start at the beginning of the year. Head or tail, bing, big R, little r, bing. Right? Right. If we would have worded this, if it's homozygous dominant, big R, 100%, right? So it's just all right. So here we go. Here's more of the the basic stuff. So this should be really quick. Chances of the offspring being a heterozygote? 50%. Okay, you do the cross, and it's kind of hard to see these, but you do the cross. 2 of them are going to have the heterozygote trait. Okay? Another one?
0.
Yeah, not possible because each parent would have to give a homozygous trait. The one parent doesn't have one. Zippo. Trick question. I know. All right. Phenotypic ratio emerges from breeding 2 heterozygotes for 2 traits, or, in other words, a dihybrid cross. So if you have 2 heterozygotes for 2 traits, pick whatever 2 traits you want. Big R, little r, big W, little w times big R, little r, big W, big W. So what is a phenotypic ratio? 9-3-3-1. Very good. Every dihybrid cross, assuming dominance for recessiveness. Okay, good question. Okay, guys, tune in real quick.
Yes.
The question was, is this always the case? And remember, there's no really always, but in dominance recessiveness, yes. But if you got something like epistasis, we pointed this out yesterday. What might happen in the case of albino, if you recall, the recessive albino traits will actually mirror or mask the expression of the black in this case. And so you're going to see a 9:3 to 4 ratio. That would be a fair game on the test because that would show that you understood that concept which we went over yesterday.
So if it's dominant and recessive, it's going to be 9:3 to 1.
Yeah.
What else could it possibly
Well, it's in a dihybrid cross, right? When you have both hybrids crossed, that would be the phenotypic ratio. So 9 of them are going to be both dominant traits, whatever they are. The 3 and the 3 is going to be, the first one will be dominant, the second one recessiveness, and this one would be the first one recessive, the second one dominant. In this case, they'll be both recessive. So whatever the trait is, remember I went over that right at the beginning when we went these dihybrid crosses. It's kind of nice to know that, just like it's nice to know the 3 to 1, so that you can see anything different than that is going to be abnormal. Incomplete dominance doesn't work that way. Codominance works differently. So you start seeing something other than these ratios, and you're saying, ooh, these traits don't express themselves normally, or they don't express themselves in a dominant recessive fashion. Okay? Yes, sir.
I think you kind of just answered this, but how would it be 9:3 4? Like instead of 9:3 1.
Yesterday, when we were going over the web quest, one of the epistasis question at the end, it showed that Punnett square with the mice.
Really?
Yeah, and in mice, it showed the expression. And then in this case, what happened was one set of genes overshadowed the normal expression and kind of dominated, and that's called epistasis, and that gave a different expression ratio than would be normal. And then in this case, there were 4 albinos out of the 16 instead of 1. Okay, and that was because of epistasis. Okay, and how would you know that? You'd have to either see it and interpret it based on what you know, or you'd have to be told there's epistasis. In that case, then you can pretty much predict what happened. Okay, but because I think I told you I'm not going to have you work a Punnett squared, but interpreting one is different. Okay. All right. Now, how many different gametes? Getting back to the Punnett squared, this is again a dihybrid, 2 hybrids, right? So, how many different possibilities are out here? This would be if you're setting up a dihybrid cross, you have to be able to do that. So work this on your mind.
16.
Ben, you know this one?
16.
Yeah. Because it makes the ones that
Okay, 4. You know what they are? Yeah, it's big. It's a foil thing, right? Bing, 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 bing.
Squaring it. I was squaring it, so I thought.
Okay, so what law does this illustrate? Which is a question you'll probably see again. Courtney? Lexi?
Sorry, what was the question?
Yes.
Law of independent assortment.
Law of independent assortment. Yeah. So you have something to suggest. All right. The idea again is these two separate independent of these two, therefore you got to look at all the possibility combinations, all the permutations, and that's what we got here. Okay. Going back, actually you could have argued that point probably, and you can too. You can say yes, you can say both, actually, right? Because in order for you to be able to independently sort them, they'd have to separate. So actually, in the dihybrid case, you actually see both. Well put. Okay? Good job. Okay, so how can you tell if anybody's home? This is going back to one of these, one of your web questions again. I think the first one, if I'm not mistaken. If you have a homozygous dominant trait, but you don't know its genotype, how can you tell? What can you do to figure it out? Go ahead. Yes.
Test cross.
Test cross. And what that is doing what with what? You can just read this.
Oh, yeah.
Yes, that's what you do. All right, and then you look at the offspring, right? And if they're half and half, you know the parent is what? The dad is heterozygous. Okay, or not the dad, the unknown parent. If they're all dominant, then you know it's homozygous dominant. Okay, so heterozygous or homozygous are pure breeding in a hybrid. You can use whatever words you want. Okay, test cross is example. Yes.
Is there going to be an essay question?
Nope. Okay, here are there's other expressions, polygenic and multiple alleles. Okay, does anyone want to take this one on?
Oh, they even got
Yeah, we did yesterday.
These are from your WebQuest, and they're also from your textbook and from the notes. Yes?
Isn't it like multiple alleles when, like, there's multiple traits, like, for one, like,
Yes.
yeah, and then polygenic is the opposite, right? Like when there's, like only one trait, and it codes, like, there's like a bunch of different codes for that one trait or something like
Perfect.
that.
Dead straight. Polygenic means take several sets of genes to basically make one trace. So basically your complex expressions, we talked about 2, skin color and height in humans. The multiple alleles basically means that you just have native expressions for one pair of alleles. So in this case, blood typing was one that we used here, and in this case I gave skin and then height. Okay? Good job. All right, what's it called when you have many genes working together to code for a single trait? Guess I should have deleted.
we just went over it.
I know I did. I should have deleted that slide if I did. Oh, oops. So you're right, polygenic. So what's epistasis? That's one of the last ones. Yes, ma'am?
One gene depends on the other.
Yes. One gene depends on the other, its expression, and it may mirror it, it may enhance it. The book showed where one was necessary to enhance an enzyme to make it work. And there's other ones that will shut one down. That's how a lot of regulation occurs in the body, actually. But in our case, we saw the albino expression masking the brown or the black allele expression. So basically, one set of genes affects the outcome or expression of it. Okay, so here we go. Eye color, sex link, Drosophila. Drosophila is a fruit fly. We kind of touched that once. Doesn't really matter what it is. So it's on the X chromosome. If the mother has white eyes and the father has red eyes, what percentage of each gender in the offspring will have each of the color eyes? So what's the phenotype? So sex linked.
Which one's dominant? Which one is recessive?
Red is dominant. Thank you.
Okay.
I don't think it's the same one.
X R, and then X big Y, Y.
So if the mother has white eyes, she's got the trait. So if we use like little r, she'd be X little r. The dad would be have red eyes, so he's X big R. So the question is, what's going to be the breakdown in each of the genders?
It's going to be 50%.
50% of males, 50% of females. Okay?
How do you tell when you cross it if they have both? It's like a big arm and a little arm?
Wouldn't it be? Wait.
Yeah, so what you got to do is we got to do a Punnett square
Yeah,
here.
it's easier to see.
This one is hard to see without. This is the same one as our colorblind one. So do you guys want to do a Punnett square real quick?
Yeah.
Wait, how would the females
Yeah, I don't even understand how you do the Punnett square.
The Punnett square, you just attach the X to the R, to the big and capital R's.
I think I can squeeze one in down here. You can see. So
Be carrots.
all right, so I got the mom, and the mom has it, right? So the dad doesn't have it.
Yeah, they would just be carriers.
Maybe not.
Yeah, I don't see how.
And the males would have it.
The males would have 100%.
All right, so if we got this, the females are going to be 0% actually, right?
0% of
And
women.
then 100% of the boys.
Red. But then it says of each color, so.
So 100% red. So what do I have on the answer here?
You add the